Hey, so in this presentation, we're going to be talking about lipids. We're talking about macromolecules or biomolecules, and lipids are a class of biomolecule. The main function is going to be long-term energy storage. They're really good at storing energy efficiently. They're a concentrated energy source, and they're easier to store. They take up less, they're not as heavy actually as storing a carbohydrate. So lipids, like carbohydrates, are going to be made up of carbon, hydrogen, oxygen. However, unlike carbohydrates, they're going to be mostly hydrogen and carbon with just a little bit of oxygen in there. So again, if you remember, carbohydrates are going to have that 1 to 2 to 1 ratio of carbon, hydrogen, oxygen. Lipids are going to have lots and lots of hydrocarbons. So here you see an example of a triglyceride. This is your kind of hydrocarbon in here. This is your hydrocarbon. So notice it's a long chain of hydrogen and carbons. The shorthand for this, sometimes you'll see it written like the zigzag. That's writing it out without the carbon. So anywhere where you see that kink, remember that's going to be a carbon with the attached hydrogens. So there's three main classes of lipids. You have your fats, phospholipids, and steroids. So fats are what we normally think of when we think of lipids. So it's like your butter, oil. Phospholipids are actually what make up cell membranes. So you've probably heard of those before. You just didn't think of them as fats or lipids. But they, you have your phosphate head and your two fatty acid tails. And then your steroids, those are things like testosterone and estrogen. Those are actually lipid-based as well. Lipids, unlike the rest of our classes of biomolecules, are not actually made up of polymers. So they're not these like this attached to this, attached to this. That's not how they're put together. They're these long chains of hydrocarbons. So they're big molecules, but they're not made of these kind of repeating subunits. So here's an example of a fat. You've got your glycerol, that's this structure right here. And that's going to be attached to a fatty acid. So you see that structure here. And the process of attaching that, remember we talked about dehydration synthesis. That's where you're going to remove a hydrogen and hydroxide with the help of an enzyme. And in that process, so you're pulling out H and an OH, so that's water. So when that comes out, you're going to leave space for a bond to be created there. So now you can actually see. So as the water molecule comes out, you're leaving that bond that needs to be created. And so these will bond with each other, and we call that dehydration synthesis. So these fats are really good at storing energy. Those long hydrocarbon chains have a lot of bonds, and bonds are energy. If you'll remember when we talked about hydrocarbons, we said that they were non-polar. So there's no overall charge here, which means that they're going to be hydrophobic. No, hydrophobic means water-fearing. So fats don't dissolve in water, which you probably already knew, but didn't think about it in these terms. Again, the main function here is going to be energy storage. They're also really useful for cushioning organs or insulating bodies. So if you think about whale blubber, that's going to be really important for insulation and cushioning. We like fatty foods because they give us lots of energy. And so we're conditioned to like sweet and fatty foods because it gives our body something that it needs. And so your body rewards you when you eat that. Eats when you eat those things with kind of happy chemicals in your brain. So when we talk about fats, there's a few different types of fats that you've probably heard of. Most commonly, we talk about saturated versus unsaturated fats. Now, you do need fats in your diet. They are important, but certain fats you need more of or less of. So saturated fats, this is where all of the carbon molecules are saturated with hydrogen. So all of their bonds are being formed and they have as much hydrogen as possible. This means that there's going to be no double bonds between those carbon atoms. And so as a result, they're going to lay really flat, long, and straight. And so they stack on top of each other really nicely. That means that they're more likely to be solid at room temperature. So most animal fats are also saturated fats. And you'll notice things like butter and lard, those animal fats are going to be solid at room temperature. And it's because of that molecular formula where they're flat fatty acids that stack nicely on top of each other. Now, these are the ones that traditionally we say you want to not have as much of in your diet. Well, butter is okay, but you don't want to go too crazy with that. Unsaturated fats, these are traditionally thought of as the healthier fats. These are going to contain double bonds between those carbons. And if you look at this image over here, you can see that where there's that double bond, you're going to form this kind of bend or kink. These are common in plants and fish. So your vegetable oils, olive oil, corn oil, those are going to be traditionally unsaturated fats. And those are liquid at room temperature. And it's because of that double bond, because of that bend in the molecule, they don't lay flat on top of each other, and so they're less likely to be solid at room temperature. When you see terms like monounsaturated and polyunsaturated, those are going to be referring to how many double bonds are found. So if there's one double bond, then that would be monounsaturated. Multiple double bonds would be polyunsaturated. When they talk about omega-3 fatty acids, those actually, it's a bend in the third carbon. Sounds really fancy when you say omega-3. So we've talked about saturated fats and unsaturated fats. You may have also heard about trans fats. So these are the ones that are actually really, really terrible for you. There is a lot of research showing that there's a high correlation between intake of trans unsaturated fats and heart disease. And so this used to be found in things like margarine or Crisco. Those were vegetable oils that were modified chemically to form what we call trans double bonds. So this is a cis double bond, and you'll notice that those hydrogens are on the same side of the double bond, which causes that bend. But if you have your hydrogens on opposite sides of those two carbons involved in that double bond, you can kind of flatten out your fatty acid, which resulted in those vegetable fats being solid at room temperature. So the problem with these is that your body was unable to process those properly, or at least the thinking goes that the reason why that resulted in a higher instance of heart disease was that those fats, your body just didn't have the enzymes to process them properly. And so they were forming these plaques, which could result in heart disease. And we'll talk more about that research and all of that later on in the year, but we're talking about fats, I think. So saturated fats, okay, in smaller amounts. Unsaturated fats are typically considered more healthy. Trans unsaturated fats, no good at all. They've actually taken those out of most foods. You don't find them in the grocery store anymore, at least in the United States. Okay, so we talked about fats, but we really haven't looked at the structure of phospholipids or steroids. These are two classes of lipids that we'll talk about extensively later in the year. Phospholipids when we're looking at cell structure and membrane structure, and then steroids when we're looking at hormones. But I did want to show you their structures while we're talking about structure. So you'll remember your phospholipid, here you go, blue. you have that phosphate head and then your fatty acid tails. So here, this would be your unsaturated fatty acid tail. And I mean, this would be your saturated and this would be your unsaturated. And you can tell because one is bent and one is not. But this is your hydrocarbon tail. And then here you have an example of a steroid or two steroids. You have estrogen and testosterone. And notice that you have these fused rings. So again, they're mostly hydrogen and carbon. So both of these, if you looked at the structure, you have those big hydrocarbon tails, which are indicative of a lipid. But they have very different kind of structures from each other. They look different. They have very different functions. But they're both made of mostly carbon, hydrogen, a little bit of oxygen. One of the clues that you're dealing with a steroid are these four fused rings. So you can see 1, 2, 3, 4. So those are your, sorry, my writing is really messy. Those are going to be indicative of those kind of steroids. So if you see that structure, that's what you're working with. All right, so let's practice seeing if we can tell the difference between a carbohydrate and a lipid. So let's look at this structure. Is that going to be a carbohydrate or a lipid? So it's made out of just carbon, hydrogen, and oxygen. The first thing I would try to do is really look at the ratio of carbon to hydrogen to oxygen. So I don't see a lot of carbon here, but there's a lot of benz with nothing on it. And so each of these is actually a carbon. And then each of those carbons, so this one is showing 1, 2, 3, that would have another hydrogen attached. 1, 2, 3, that would have another hydrogen. Sorry, I will try to write neater. This one has 1, 2, 3, it has another hydrogen. This one has 1, 2, 3, it would have another hydrogen. This one is 1, 2, 3, oh, and then it's got the OH to it. Okay. So I think I've written them all in. Let's count it up. So I've got 1, 2, 3, 4. Oops, I forgot about this guy. 5, 6 carbons. So this is C6. Now let's count the hydrogens. So I've got 2, 3, 4, 5, 6, 7, 8, 9. Am I doing this right? 10, 11. Where am I missing one? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, I just can't count. H12. All right, now let me see oxygen. So I've got 1, 2, 3, 4, 5, 6. O6. So I've got a 1 to 2 to 1 ratio of carbon, hydrogen, oxygen. That's going to be indicative of a carbohydrate. This is actually glucose. Another thing to look for in the name of a carbohydrate, your sugars usually are going to end in that ose. All right, let's look at the next one. Carbohydrate or lipid? Well, I've got a whole bunch of carbons, a whole bunch of hydrogens, and then 1, 2, 3, 4, 5, 6 oxygens. So I don't have even close to a 1 to 2 to 1 ratio of carbon, hydrogen, oxygen. And I have these long chains of hydrocarbons, so carbon with hydrogens attached. So I'm going to go lipid on this. This is a triglyceride. So now let's look at those fatty acids. Would this be saturated or unsaturated? So if you look, all of those carbons are full of bonds. It's straight. So this would be a saturated. And if we look at this one, we have that double bond between those two carbons, which is causing the bend. That would be a good example of an unsaturated. And I apologize for my really terrible handwriting on the screen. All right, this one's a little bit trickier. What's going on here? So if I look, I've got 1, 2, 3, 4, 5, 6 carbons. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 hydrogens. Now let's look at oxygen, because that's really what's going to tell me. 1, 2, 3, 4, 5, 6 oxygens. So this is actually a different way of representing glucose, the same molecule we saw a couple slides ago. Be careful, because it could be represented in both ways. And they're both the same molecule. They're just different ways of drawing it out. All right, time for a break.